Guys, did Dr. Aisha and Dr. Ali had share about the tips for our biochem paper?
Yeah, they did. It was on our last class.
Can you tell me what is it?
If I am not mistaken, it's about Kreb cycle and also etc.
Oh my god, I am not really understood that part.
It's okay, I will teach you about that, okay? So, the Kreb cycle is a series of chemical reactions used by all aerobic organisms to generate energy through the oxidation of acetate, derived from carbohydrates, fats and protein into carbon dioxide. Theoretically, there are several alternatives to the TCA cycle, but the TCA cycle appears to be the most efficient. The citric acid cycle consumes acetate in the form of acetate-CoA and water, reduce NAD plus to NADH and produce carbon dioxide. Citric acid cycle is an alternative name for the Kreb cycle. The cycle provides precursor including certain amino acids as well as the reducing agent NADH that is used in numerous biochemical reactions. Its central importance to many biochemical pathways suggests that it was one of the earliest established components of cellular metabolism. It may have originated biogenically. So next is electron transport chain. As I said before, the Kreb cycle, it generates ATP. But most of the ATP generated during the aerobic catabolism of glucose is not generated directly from this pathway. It is derived from a process that begins with moving electrons through a series of electron transporters that undergo redox reduction. The process is called the electron transport chain. This causes hydrogen ion to accumulate within the matrix space. Therefore, a concentration gradient forms in which hydrogen ion diffuse out of the matrix space by passing through ATP synthesis. The current of hydrogen ion power the catalytic action of ATP synthesis which phosphorylate ADP producing ATP.
Oh, I see. So how the Kreb cycle works?
Kreb cycle begins after the process of glycolysis and before the ETC. Glycolysis will produce pyruvate as its end product. From the pyruvate oxidation process, it will produce acetyl coenzyme A, or known as acetyl-CoA, and carried out by pyruvate dehydrogenase complex. The conversion of oxaloacetate from pyruvate is carried out by pyruvate carboxylase when abundant of acetyl-CoA. So, overall cycle for the Kreb cycle, it has 8 steps. Acetyl-CoA joins oxaloacetate and forms citrate with the assistance of citrate synthase. Then, citrate converts into its isomer, which is isocitrate by dehydration-hydration reaction with the aid of acetonitase. Then, the isocitrate will oxidize and release carbon dioxide. At the same time, NAD plus reduce to form NADH, so it will form alpha-ketoglutarate. This reaction comes with isocitrate dehydrogenase, which to regulate the speed of the Kreb cycle. In the fourth step, alpha-ketoglutarate transforms to succinyl-CoA by enzymatic decarboxylation and oxidation. The enzyme catalyzing is alpha-ketoglutarate dehydrogenase. During reaction, it releases carbon dioxide and NAD plus reduced to form NADH. In the fifth step, reaction involved by the succinyl-CoA, ADP and phosphate with the enzyme succinyl-CoA synthase. The hydrolysis of this metabolite to succinate coupled to the phosphorylation of guanosine diphosphate or GDP to guanosine triphosphate GTP. The CoA in succinyl-CoA replaces by phosphate group and it will transform ADP to ATP. In the sixth step, enzymatic desaturation by a flavin adenine dinucleotide FAD dependent succinate dehydrogenase. Succinate oxidize and form fumarate. In these reactions, two hydrogen atoms with their electrons are transferred to FAD and forms FADH2. The enzyme at step 6 is embedded in the mitochondrial inner membrane. As a result, FADH2 can transfer its electron directly into the electron transport chain ETC. After the hydration, the fumarate catalyzed by fumarase and transforms into malate. In the eighth step, Malate to oxaloacetate is catalyzed by malate dehydrogenase. Hence, NAD+ reduced to form NADH. As it reaches at step 8, the cycle will repeat and repeat again. For your information, Kreb cycle takes place in all eukaryotic and prokaryotic cells. In eukaryotes, it occurs in the matrix of the mitochondrion, while in prokaryotes, it takes place in the cytosol. The application of Kreb cycle are lipid metabolism, amino acid metabolism, protein metabolism, glucose metabolism, energy expenditure, and also metabolomics.
Wow, so interesting. Then, electron transport chain, or also known as ETC, will take place after the Kreb cycle. The ETC is located in the mitochondria of a cell with the function to produce ATP. The ETC consists of a series of oxidation-reduction reactions that lead to the release of energy. A summary of the reaction in the electron transport chain is as pictured, where H2O and ATP are the end product. There are 4 complexes in ETC, starting with complex 1. Complex 1, also known as NADH dehydrogenase, pumps 4 hydrogen ions from the matrix into the intermembrane space, establishing the proton gradient. In the next protein, complex 2 or succinic dehydrogenase, succinic is oxidized into fumaric, causing FAD to be reduced to FADH2. Then, FADH2 transfers electrons to complex 2 and the electrons are passed along to ubiquinol. Q is reduced to ubiquinol, which carries the electron to complex 3. No hydrogen ions are transported to the intermembrane space in this process. The passage of electron to complex 3 drives the transport of 4 more hydrogen ions across the inner membrane. Ubiquinol is oxidized and electrons are passed to another electron carrier protein, cytochrome C. Cytochrome C passes electrons to the final protein complex in the chain, complex 4. 2 hydrogen ions are pumped across the inner membrane. The electrons are then passed from complex 4 to an oxygen molecule, causing the molecule to split. The resulting oxygen atoms quickly grab hydrogen ions to form 2 molecules of water. Finally, ATP scientists move the hydrogen ions that were pumped out of the matrix by the electron transport chain back into the matrix. The energy is used to generate ATP by the phosphorylation, or known as addition of a phosphate of ADP. The movement of ion across a slightly permeable mitochondrial membrane and down the electrochemical gradient is called chemiosmosis. Oxidative phosphorylation will take place after electron transport chain. It is also the final stage of cellular respiration.
Now I'm really understood those cycles. So it can be concluded that the glucose is completely broken down, yet only 4 ATP have been produced. Moreover, although oxygen is required to drive the Kreb cycle, the cycle chemical reaction do not themselves consume the oxygen. The end product of the ETC are water and ATP. A number of intermediate compounds of the citric acid cycle can be diverted into the anabolism of other biochemical molecules, such as non-essential amino acids, sugars, and lipids. Am I right?
Yes, sure, you are right, Is.
Yeah, now I'm ready for the paper tomorrow.